Now, DNA then has all of the genetic information, but how does that genetic information make the phenotype, right? Well, the phenotype, as we've stated before, are all of the specific traits that were coded for by the genotype. And so you can think about it in this way. The genotype leads to the phenotype, right? We used to say, big P, little p, or big P led to purple flowers. But we can also break this down at a molecular level and say DNA leads to RNA, which leads to eventually polypeptides or proteins. And I just want to remind you that we're not forgetting that the environment is also an important part of determining the ultimate phenotype that is produced. But for the meantime, we're going to ignore the environmental effects. So here's a close-up of a cell where we're looking at the nucleus here and then the cytoplasm on this side. DNA is housed inside of the nucleus in eukaryotes, and the DNA never leaves the nucleus. So DNA goes through a process called transcription to create an RNA copy of a gene. This RNA copy we call a messenger RNA. This RNA molecule can leave the nucleus through the pore and come outside into the cytoplasm where it can undergo the process of translation and then eventually become a protein. So here's where we're going from DNA to RNA to protein through two processes, transcription and translation. We're going to look at both of those in detail. So if we look at this even kind of zoomed up a little bit, so here is some DNA, here is a gene, and we zoom up to look at the actual nucleotides. What's really happening then is you're going from a DNA strand, you're copying and getting the RNA complementary base copy of this. So A's bind with U's. Remember in RNA we have U's instead of T's. C's bind with G's, and it doesn't show this here, but a T would bind with an A. And the message that is encoded in the genetics is written in 3-letter triplets of bases. We call these codons. And each codon codes for one amino acid. So the process of transcription is going from DNA to RNA, and the process of translation is going from RNA to a polypeptide, where you translate from nucleotide language into a different language, which is written in amino acids, and then eventually becomes a polypeptide or a protein. A number of scientists figured out the genetic code, and this was a really important hurdle that we made in science, where we started to figure out which triplets or which codons code for particular amino acids. So UUU, if you have an RNA molecule and it has a UUU, then you know that's going to code for phenylalanine, as will UUC, but UUA will code for leucine. So look at this base. As you can see, there are 64 possible bases, because if you have 3 different nucleotides forming one codon, and you have 4 different nucleotides that can be put into that triplet, then there are 64 different possible combinations. You can see along here we have the first base is indicated by the column, by the row, the second base is indicated by the column, and the third base is indicated by the rows within each of these boxes. And as you look across here, notice that you see some patterns forming. First of all, you see a start codon, that also is the same codon as methionine, and you might imagine that that has something to do with the start of the genetic message in any gene, and that's true. You also have codons that are called stop codons, and as you might imagine, those codons tell your body that that is where the genetic message ends. That's where the gene ends. And then you have all the rest of the amino acids. There are 20 total amino acids that are shared across life, with a few exceptions. And these amino acids also have patterns. So look at, and you can see like here in threonine, you see that it doesn't matter what the third position nucleotide is. As long as it's A, C, something, it's going to be a threonine. And that is very common for many of these. Or you might have it where a CAU or CAC is a histidine, or AAU or AAC is an asparagine. And you see these patterns forming. Now notice that if you there's a few times when you can actually change the first position and still maintain the first amino acid. For example, down here, CUA is leucine, but if the C changes to a U, UUA, it's still leucine. That's also true over here for arginine. AGA to CGA is still arginine. However, there is never a case where you can change the second position, the second nucleotide position in a triplet, and still maintain the same amino acid. It does not occur. Now, what is interesting is that this genetic code is shared by all organisms. And so this is why you can take a gene out of one organism, like this firefly, and put that gene inside of a tobacco plant, and it can function and it can work. Genes can be taken out of one organism, put in another organism. We actually do this quite often, where we take human genes that are of interest and put those into bacteria, and the bacteria will make the protein product from that gene. This is a really common method that we now use to make human insulin, and that is then given to diabetic patients. So the process then of doing this, remember, was divided into transcription and translation. And during transcription, there is another important molecule that I want you to know, and this is called RNA polymerase. RNA polymerase also unzips the DNA molecule, breaks apart the double strand, and then it leaves open a side of DNA to become the template strand. And as the RNA polymerase runs down the strand of DNA, it then makes a copy of that gene in RNA. And so you end up with this newly made messenger RNA. So the way that it does this is the RNA polymerase sits down just upstream of where the gene is. So this is before that, what we call the start codon, and even a few nucleotides before that, upstream, the RNA polymerase sits down on an area of the DNA called the promoter site. Then it moves down and finds and starts to make a copy of the message that includes a little bit of the DNA just above the start codon and goes all the way down and it includes a little bit of DNA even beyond the stop codon. Then it hits a region called the terminator DNA, and that is when the RNA polymerase knows that it now is done and it lets go of the DNA strand, and you have a completed RNA strand. The three words that we use for this process is initiation, where it binds and starts to move, elongation as it goes away, all the way along the gene, and termination when it finally lets go of the DNA. And that's the process of transcription. Now in eukaryotes, there's one other thing that needs to happen, and that is called RNA processing. So here's the DNA. We go through and do the transcription and we get this RNA product. But notice that in eukaryotes you have different parts of the genetic message called exons and introns. These were named by Walter Gilbert in '78. Intron refers to the intragenic region, so the regions between the actual genes, or between the information that is really genetic information that's going to be used to make the protein. So it's as if the introns are just spacers, just fillers. And the exons are the actual expressed regions. These are the regions that are going to be expressed. So as you might imagine, we don't want the introns. So after you make the copy of RNA, the introns are removed, and then the exons are spliced together. Spliced in molecular biology means pasted together. And now you have the coding sequence all put together as it should be put together. You also add caps and tails that help protect the strand and also allow the binding of the ribosome to happen. And then now it's ready to go outside of the nucleus. Just as an aside right now, I want to talk about something that's really important, and this is called alternative splicing. It turns out that in eukaryotes, you can actually sometimes splice together exon 1, exon 2, and exon 3, or just exon 1 and exon 2, or just exon 2 and exon 3, or just exon 1 and exon 3. And so you can have these alternative ways of splicing together these exons. And this is why you, even though humans only have 20,000, a little over 20,000 genes, we can make more than 200,000 different kinds of proteins because we can alternatively splice the different exons together to make slightly different proteins. And this was a huge discovery just made in the last 15 or so years. The next part of the process then is that once that messenger RNA leaves the nucleus is then to undergo the process of translation. So the ribosomes are the sites where this occurs. And so I just want to remind you of the structure of a ribosome. It has a small unit and a large subunit. In the large subunit, there's what's called the P site and the A site, and this will come into play. You'll see that this is where these transfer RNAs, a different molecule that we have in our bodies, come and dock in these different sites. And then we also have this groove that comes through here, and this is where the messenger RNA is attached. So let's put it all together. Here we have the ribosome in blue, the messenger RNA that we just made through the process of transcription, and then, as I said, we have these two docking sites, and this is where you have these transfer RNAs. So these are RNA that fold up on themselves, and they have two important parts. On one end, they have an amino acid attachment site, and on the other hand, they have what's called the anticodon. And as you may guess, the anticodon has the corresponding bases to attach to the codons, right? And we'll see that in just a moment. And so when it's all put together and working, you basically have from the P site, you have the growing polypeptide, and then in the A site, you have the next amino acid that's going to be added to this growing polypeptide. So let's start this off. And again, they use the same words to talk about this of initiation. So in initiation, everything comes together. The ribosome grabs onto the messenger RNA, and the first tRNA is brought into place, and we know that the first tRNA that is brought into place is always the same tRNA. It's the start codon. Remember, that was methionine, and that is AUG. So the anticodon that is part of the tRNA would be UAC. In every case, that's how it is. Okay, so now we'll move on to the next part of the process here, where we'll see what will happen now that the A site is available. It's ready to receive the next transfer RNA to bring in, that's going to have the corresponding anticodon, and so therefore it's bringing in the correct amino acid. And so that occurs, and then you go into what's called translocation, first the peptide bond formation. So once that site gets filled, then you form a peptide bond, which is a special type of covalent bond between these amino acids. Then the P site lets go of now the uncharged tRNA, so this tRNA that now does not have this amino acid. And then you have translocation, where everything shifts down, and the growing peptide chain now goes into the P site. Now the A site is ready to receive the next amino acid. And this just keeps chugging along. You know, it's kind of like pulling, remember a tape player that would pull that magnetic tape through the magnets, and you could read the message and basically play music. Well, that's kind of what's happening. It's pulling this message through, and at the same time, it's making this growing polypeptide. And that continues until you hit the stop codon. So a basic review then of this whole process is, once again, you start with the DNA, the RNA polymerase comes in, copies, makes the messenger RNA, that RNA is then processed, exons are spliced together, it comes outside, runs into the ribosome. You start with the very first start codon, and then you continue to do this process again and again and again until you're done with the polypeptide. Once you hit the stop codon, the ribosome knows to let go, and the polypeptide is now fully formed. In the meantime, by the way, off to the side, these tRNAs that are being used are called uncharged, and then an enzyme goes through and reattaches the correct amino acid back to that tRNA, and it becomes charged, and then it can be reused again. So that's the entire process of transcription and translation.